Hello everyone, I am Dr. Geeta Hegde, and in this series of videos we will be learning about the various actions of insulin. In this particular video we will be learning about metabolic effects of insulin on carbohydrate metabolism. Insulin is a crucial hormone in regulating carbohydrate metabolism with several key effects that help maintain blood glucose levels within the normal range. The primary metabolic effects of insulin on carbohydrate metabolism are increased glucose uptake, promotion of glycogen synthesis, inhibition of glycogenolysis, suppression of gluconeogenesis, enhanced glycolysis, and lastly, inhibition of lipolysis. Insulin is the most important hormone for blood glucose regulation. It is a primary hypoglycemic hormone which reduces the blood glucose levels by negative feedback mechanism. It is released from the beta cells of pancreatic islets. The first effect of insulin on carbohydrate metabolism is promotion or increased glucose uptake. Insulin stimulates glucose uptake in muscles and fat tissues by promoting the translocation of glucose transporter type 4, GLUT4 receptor to the cell surface, and this facilitates glucose entry into the cell. So increased glucose uptake is by facilitating translocation of GLUT4 receptors to the cell surface, and this promotes glucose entry into the cell. The mechanism of glucose uptake occurs by first step is insulin binding. Insulin binds to the insulin receptors on the surface of target cells, primarily skeletal muscle cells and adipocytes. The second step is signal transduction. The binding of insulin to its receptor triggers a signaling cascade inside the cell. This involves activation of insulin receptor substrates and phosphatidylinositol 3 kinase. The third step is activation of protein kinase B, which is also known as AKT. This enzyme plays a key role in glucose uptake. The fourth step is translocation of GLUT4 receptors. Translocation of GLUT4 receptors, which is glucose transporter type 4 vesicles, to the cell membrane. GLUT4 is the primary glucose transporter in muscle and adipose tissue. Once at the membrane, GLUT4 facilitates the uptake of glucose into the cell by allowing glucose to pass through the cell membrane down its concentration gradient. Importance is increased glucose uptake provides cells with glucose needed for glycolysis. Second, it promotes glucose storage. And third is adipocytes convert this glucose into triglycerides, contributing to fat storage. The second step of insulin on carbohydrate metabolism is increased glycogen synthesis. This happens in 3 steps. Insulin binds to the receptors on cell and muscle cells and activates the insulin signaling pathway, similar to that of glucose uptake. Second step is activation of glycogen synthase, which promotes storage of glucose and converts it into glycogen and helps in storing this glycogen. The third is inhibition of glycogen synthase kinase 3. The third step of insulin on carbohydrate metabolism is inhibition of glycogenolysis. Glycogenolysis is breaking down of glycogen into glucose by reducing the activity of enzymes like glycogen phosphorylation or glycogen phosphorylase. Mechanism of action remains the same. Insulin binds and initiates signal transduction. Second step is activation of protein kinase B. Third is inhibition of glycogen phosphorylase. Fourth step is decrease in the cyclic AMP levels. These lower cyclic AMP levels decrease the activation of phosphorylase kinase and glycogen phosphorylase. And this decreases the activation of breaking down of glycogen, which is known as glycogenolysis. The next step is suppression of gluconeogenesis. Insulin suppresses gluconeogenesis. What is gluconeogenesis? Gluconeogenesis is the production of glucose from non-carbohydrate sources like amino acids in the liver. Insulin plays a main role or a crucial role in suppressing gluconeogenesis. The process of producing glucose from non-carbohydrate precursors such as lactate, glycerol and amino acids. This takes place in liver. This suppression is essential for maintaining the blood glucose levels within a normal range. The suppression of gluconeogenesis again occurs in the following steps: activation of protein kinase B, inhibition of PEPCK, which is phosphoenol pyruvate carboxykinase. The third step is inhibition of glucose 6 phosphatase. Fourth is enhancement of glycolysis, inhibition of fructose 1,6 bisphosphatase. Thus, insulin promotes glycolysis, which is again the fifth step. How does it promote glycolysis? Enhanced glycolysis. Insulin stimulates glycolysis, which is breaking down of glucose to produce energy. This enhancement of glycolysis is done by activation of two enzymes. One is phosphofructokinase and second is pyruvate kinase. The sixth mechanism is inhibition of lipolysis. While primarily this is fat metabolism, insulin is involved in inhibition of lipolysis. Insulin helps reduces the availability of glycerol for gluconeogenesis, and this is the indirect effect on carbohydrate metabolism. So overall, insulin acts to lower blood glucose levels by promoting the storage and utilization of glucose while inhibiting its production and release, thus maintaining energy homeostasis. To summarize the effects of insulin on carbohydrate metabolism, the first step is promotion of increased glucose uptake, which is through the help of GLUT4 receptors which promote glucose uptake by the cells. The second step is promotion of glycogen synthesis. The third step is inhibition of breaking down of glycogen, which is known as inhibition of glycogenolysis. The fourth step is suppression of gluconeogenesis. Fifth is enhanced glycolysis, and last is inhibition of lipolysis, which is an indirect effect of insulin. Thank you.